SOLUS is an innovative tomographic system that combines diffuse optics and ultrasound imaging to support the diagnosis of breast cancer, the most common cancer in women in Europe and worldwide. The main goal of SOLUS is to improve the discrimination between malignant and benign breast lesions by characterizing breast tissue non-invasively, thus reducing unnecessary biopsies and further invasive examinations for the patient quality of life and for the economic sustainability of the healthcare systems, as a huge number of needless examinations are currently being performed.
To reach its goal, SOLUS has developed a multimodal handheld probe combining B-mode ultrasound imaging, shear-wave elastography, and diffuse optical tomography to estimate in a single measurement session tissue morphology, stiffness, and composition. This has required the development of groundbreaking technologies made possible only through the combination of different expertise, photonics, electronics, medical imaging, radiology, and through the continuous collaboration of all partners. Great efforts were made to design the miniaturized components for the optical measurement with state-of-the-art and even better performances. From the laser driver to generate picosecond pulses at different wavelengths in the red and near-infrared, to the high-sensitivity wide-area time-gated detector, to the dedicated integrated acquisition electronics. All components were then integrated in a single probe with the ultrasound transducer, with cost-effectiveness always in mind. The management of a complex system and the 3D reconstruction of collected optical data required the design of dedicated algorithms and custom-made software. Performance assessment of the developed instrument is key for its successful use. For that, new protocols and tissue phantoms suitable for bimodal ultrasound and optical imaging were developed and applied. SOLUS was eventually transferred to the clinics for validation in real settings on patients with breast lesions. The typical SOLUS exam begins with an initial measurement on a breast tissue phantom to check the instrument performance. Then the patient is welcome and the radiologist illustrates the clinical protocol. If the patient provides or informed consent, the examination can start. The entire exam is performed in dim light for easier examination of the ultrasound images and to avoid the interference of room light with the optical detectors. For this initial trial, the radiologist is always supported by an engineer so that we can learn about strengths and weaknesses both on the medical and the technical side. First, the radiologist identified the lesion with a standard ultrasound probe. Then the measurement with the multimodal SOLUS probe can start. Each acquisition includes B-mode ultrasound imaging, shear-wave elastography, and tomographic optical data. The procedure consists in 4 acquisitions in different positions: along the main axis of the lesion, along the orthogonal axis, far from the lesion in the same breast, and at a mirror position in the contralateral breast. This is needed because optical assessments are based on the contrast between lesion and healthy tissue. Moreover, different views enable a better characterization of the nodule. The measurement procedure is repeated by 3 different radiologists to evaluate whether the results are independent of the operator and to provide us with more feedback on the usability of the system. At the end of the exam, data are analyzed in real time. The lesion is identified on the ultrasound image, thus providing morphological a priori constraints to the optical reconstruction and significantly improving localization and quantitation of the lesion composition. In few minutes, 3D maps are obtained of the hemoglobin, lipid, water, and collagen contents of the investigated breast volume. The optical tomography can be scanned along 3 orthogonal sections and depicts the lesion in different color where SOLUS recognizes a mismatch in the lesion composition with respect to the surrounding tissue. The results are then discussed with the radiologist. Further refined analysis is also performed offline for improved quantification.
The clinical validation of SOLUS is currently ongoing and will continue in the next month to test the potential of the SOLUS approach in diagnosing breast cancer and to identify possible improvements in other applications with the contribution from all partners. If you are interested in the system and its use, get in touch with us.